Angelman Syndrome Genotypes, sponsored by the Foundation for Angelman Syndrome Therapeutics. Everyone inherits their chromosomes from their parents. The chromosomes carry genes, which are coding instructions for making proteins. An important example of this is the UBE3A protein, which is made from the UBE3A gene located on chromosome 15. People living with Angelman syndrome, or AS for short, do not have enough functional UBE3A protein in their brains. Typically, each person has 2 copies of the UBE3A gene, one from each parent. The one from the father is called paternal, and the one from the mother is called maternal. In all people, the paternal UBE3A gene is silenced or turned off in brain cells, a process called imprinting, so the maternal UBE3A gene is responsible for making all the UBE3A protein in the brain. Now, changes to the maternal UBE3A gene can result in less or no UBE3A protein in the brain, or the UBE3A protein that is produced may not work properly. These genetic changes are grouped by type and are called genotypes. There are 5 main genotypes that cause AS. Knowing the genotype of each person living with AS is important. Although each individual living with AS is unique, people with the same genotype may have certain behaviors or characteristics in common. Another reason for identifying the genotype is that some genotypes have a higher chance of running in families. Doctors and genetic counselors can use this information to determine the chance of other family members inheriting AS. Finally, genotype is often a criteria for participating in research, so knowing the genotype is a key element in determining eligibility for clinical studies investigating potential AS therapies. Mutation AS occurs in about 10% of people living with AS. In this genotype, a coding change, also called a mutation or variant, within the UBE3A gene causes a change in function of UBE3A protein, making it inactive or less active. Mutations can occur anywhere in the gene and are categorized based on how they affect the gene and the protein. Most UBE3A mutations result in no UBE3A protein, but some may allow for some protein to be made, although that protein is likely less functional or may be completely non-functional. Individuals with some functional UBE3A protein may have fewer or less severe AS symptoms. Unlike the other AS types, which use DNA methylation testing for diagnosis, mutation AS is diagnosed by sequencing, which is a test that reads genetic code. Mutation AS may be inherited from the mother who has the same mutation, or it can be a brand new change in the egg, also called de novo, that made the person living with AS. If the mother has the mutation, each future pregnancy has a 50% chance of inheriting mutation AS. For more information about Angelman syndrome and its genotypes, visit the Foundation for Angelman Syndrome Therapeutics, also called FAST, at cureangelman.org. For specific questions, reach out to FAST's genetic counselor.